All right, hey guys, let's talk about nucleic acids today. So nucleic acids. Now to start with nucleic acids, you know where we like to start. We like to start with the elements. So here's my elements. The one element that everything has to have inside of it is carbon. And then the other two elements that we always see are hydrogen and oxygen. And also, nucleic acids have N, which is nitrogen, and P. That word doesn't really spell anything, so I usually like to rearrange those and make that as punch, okay? So these are the elements that are found inside nucleic acids. If we look at the monomer of a nucleic acid, the monomer of a nucleic acid is a nucleotide. And they sound really, really same. So we have nucleic acid, and then we have nucleotide. Now my nucleotide is made up of 3 different things. 1, 2, 3. Now remember, we always like to use the elements to start building our monomers. So if I look at my first element, this is the P, and the one P that we've talked about is a phosphate group. Then the other elements that we've looked at, CH and O, that will give us a sugar. I'm going to put an asterisk next to that because we're going to talk about it a little bit more. And then the last one, we'll get our last element on there. This is going to be a nitrogen base. And now that we've listed those out, we can draw them. So phosphate group, let's draw that first. So here's a phosphate group. Remember, super negative. Got negatives everywhere. And now we get to draw a sugar. So our sugar is going to look something like this. We've talked about how we can draw sugars to look like that because we can look at carbons. And if I look at that sugar, how many carbons does this sugar have? It has 1, 2, 3, 4, and 5. Since it has 5 carbons, we can look at our alkanes and do the word root. That's pent. And then since it's a sugar, I use the suffix. This is a pentose sugar. And a few things coming off that pentose sugar right here at the 3 carbon and right here at the 2 carbon. That's going to change depending on what type of nucleic acid we're looking at. And then my last part, the nitrogen base, we're just going to draw that as NB. So we'll talk more about structure and stuff later. All right, so now that we have the elements and we have the nucleotide, the monomer, and then the pieces of the nucleotide, now we can talk about examples of this. So my examples of a nucleic acid are DNA and RNA, because that's where we get the names from. So DNA stands for deoxyribose nucleic acid. That's the DNA, deoxyribose nucleic acid. And then RNA stands for ribose nucleic acid. That's where the RNA comes from, RNA. So now that we know what that stands for, we can start kind of doing some differences between them. If I look at this first word, deoxyribose versus ribose, we should know what molecule that's referring to because it ends in ose. The thing that's referring to is the sugar. So the sugar in DNA is deoxyribose versus the sugar in RNA is just ribose. And what that looks like, how that's different, is right here when we looked up at this sugar molecule and we put these 2 question marks. If this is a ribose sugar, these question marks aren't question marks anymore, but your question marks get to be a hydroxyl group because then that's just what ribose looks like. But if we think of the word deoxyribose, well, think about what D means. D means not or un, and oxy is referring to oxygen. And so when we look at my deoxyribose sugar over here, this one right here doesn't exist because DNA is deoxy, or it doesn't have that oxygen there. Ribose would have both of them. So that's just a structural thing. And then the NB stands for nitrogen base. The nitrogen bases are different. So NB, nitrogen base. Nitrogen bases in DNA are adenine and thymine, cytosine, and guanine. Those are the 4 nitrogen bases here. And then in RNA, the nitrogen bases are adenine, uracil, cytosine, and guanine. And so if we look at this list, we can see where the difference is. DNA has a thiamine and RNA has a uracil. And so that's where those are different in their nitrogen bases. Now, nitrogen bases, little segue, we need to talk about them. When we think about the nitrogen bases as a whole, they always have base pairing rules. These are called Chargaff rules, and we'll go through it. We'll go through it more so when we get to that unit. But nitrogen bases, they always pair up together. And when we look at these, say A's and T's go together, and C's and G's go together. And I always have a dumb way to remember. I always say that apples grow on trees and cars go in the garage. So when we look at my DNA molecule, that adenine, that adenine and thiamine pair up, and then your cytosine and guanine pair up. Versus on RNA, if I'm looking at these, the nitrogen bases, my cars still go in the garage. That's the one that's the same throughout. But now instead of my apples being on trees, my apples fall under the trees. So C and G still goes together. That's the same between the nitrogen bases on DNA and RNA. But now apples fall under the tree, and that's going to be different right there. Now, nitrogen bases, something else we need to note. Nitrogen bases, we actually have 2 kinds. The first kind, this is called a purine, and a second kind is called a pyrimidine. Now, a purine, structurally, what makes it different is that there are 2 rings here. In pyrimidine, there is 1 ring here. Now the purines, the nitrogen bases that fall under this, are A and G, and then pyrimidines are going to be the C's, T's, and the U's. And so really what that looks like whenever we do a DNA molecule, we've drawn these before, they always kind of look like a ladder. So I always have 3 rings right here in the middle because A's would fill up 2 of them, and since it pairs with T is only 1. Versus if I have a G, so maybe this is G right there, and then that would pair up with C, which is 1. Something to note, we'll talk more about it later. Now, to finish this up over here, that's pretty color. To finish this up over here, we need to do function. So the function of DNA, and we've done function before. Function of DNA is to store genetic info, which makes sense because if I ask you guys, hey, I'm going to draw a cell, first, how do I draw a cell? You're going to tell me you draw a circle. And since it's one of our cells, we're going to draw a nucleus. And then inside our nucleus, this is where we have our DNA. Now, what maybe we haven't learned before in other classes is that this nucleus is full of holes. Okay, these are called nuclear pores. Now, to go back to this, DNA, we know it's you know that DNA stores genetic information. But what is that genetic information actually doing? It's building a living organism. And what's the thing that builds living organisms? Well, those are proteins, right? Proteins, we've always said, is going to build a physical organism. All right, well, if I have proteins, that means I need to make them. How do I make them? I make them at the ribosome structure, because ribosomes, what they do is they link amino acids. But the catch is, all the way out here in my cell, this is where the ribosomes are. They're outside the nucleus. So what I need to do is get this code that's here in the DNA, and I need to get it over here. But if I look at my DNA, because it's two-stranded like this, he's too big to fit through the holes. So what do I have to do is I'm going to make a copy of the DNA. So maybe this yellow guy, he makes a copy of that DNA, but look, since he's single-stranded, he's going to be able to go out that way. And so if I add that to my list, DNA, if we're talking about the number of strands, DNA has 2 strands. Whoops, let's spell strands right. He has 2 strands, versus my RNA only has 1 strand, which gives us RNA's function. RNA's function is to transfer genetic info to the ribosome. And that's why we have both of these nucleic acids, because the DNA over here in this picture, he's too big to fit through the holes, but the RNA can, and so then that RNA can take that genetic code, a copy of it, to the ribosome, because then once he goes to the ribosomes, he's going to be able to link those amino acids so he can build a physical organism. And I think that's about all we need to know right now for our DNA and our RNA nucleic acids.